Mhm. Good afternoon. Welcome to World Forum for Women in Science 2024, Envisioning Tomorrow, and we are in the technical session right now. And I would like to give the word to our moderator for today to introduce the first speaker. Thank you. Dear Dr. Olanike, does it work for you with the microphone? Yes.
Hello, good afternoon, everyone. Are we here together? So good afternoon. Can we all see me?
Yes, we can see you and we can hear you.
Yes. I want to welcome everyone to this session. Can we start now? Please, can you hear me?
Yes, we can hear you, and go ahead to start the session and to introduce the first speaker.
Okay. Good afternoon once again, everyone, and I would like to welcome us to this session on health. I would like to specifically appreciate the organizers for the opportunity to feature on this platform. And most importantly, I want to say very big thanks to Nerina for this singular honor given to me. And I want to say I do not take it for granted. This session is on health and is one of the 17 sustainable development goals known as the global goals agreed by the members of the United Nations to accomplish in the year 2030, and is a universal call to action to achieve peace and prosperity by the year 2030. So each of these 17 goals are very crucial and important, and as crucial as they are needed for sustainable growth for all the continents, not particularly to a particular continent, but to all. And at this, so it is crucial for us to put heads together to contribute a quota to discussions that will help in achieving these sustainable development goals. And central to this, to all of that session, I believe this the insight we draw from this session will provide insight and necessary contribution in achieving the SDG 3, which is on health. So in this session, I believe we have five presenters. I don't know if everybody is available. Let me check. We have 5 presenters. We have Dr. Brenda Wanjiru. Is Dr. Brenda Wanjiru available today? Then we, yes, okay, thank you. Then we have Dr. Poja.
Yes, yes,
Thank you. We have Dr. Temiloluwa Afolayan. Is she available here?
No, unfortunately.
Okay. Probably she will join us soon. Then we have myself. I happen to be one of the moderators and also one of the presenters. And also we have Dr. Ayasama Mohammed. Is she available? Okay. Now I would like to to give the platform over to Dr. Brenda. Dr. Brenda is a molecular biologist and bioinformatician at Jomo Kenyatta University of Agriculture and Technology. I think that is in Kenya. And she's working, she's going to discuss structural and functional implication of MIT2 and NT2 mutations in hamodiaquine and piperaquine resistance Plasmodium bengae parasite. I would like to introduce her in a brief introduction. She's a molecular biologist and bioinformatician based at Jomo Kenyatta University of Agriculture and Technology Institute of Biotechnology Research. She has garnered substantial experience in biotechnology and her contribution extends to peer-reviewed publications and successfully securing funding for a research project. Our work has enhanced the comprehension of molecular mechanisms involved in parasite or drug resistance. And she has successfully identified potential drug targets and resistance mediators, and which includes hands-on experience in transfection work, utilizing plasmodium and germ knockout vectors for Plasmodium bengae, adding in the validation of drug target and resistance markers. As key set also encompasses the proficiency in R and experience with Oxford Nanopore technology for sequencing, coupled with command line expertise in data analysis. So we welcome you on the platform, Ms. Brenda Wanjiru. You have 12 minutes to present your presentation. Thank you. So, Ms. Brenda?
Yes, I'm currently sharing my screen.
Yes, you can try sharing. Yes, thank you. Yes, we can see your screen.
Hello, everyone. My name is Brenda Murithi, and I will be taking you through the structural and functional implications of MIT2 and NT2 mutations in amodiaquine and piperaquine resistant Plasmodium bengae. I'll start with a brief introduction of what malaria is. It's a disease caused by a parasite named Plasmodium, and it's spread through a bite from an infected female Anopheles mosquito. The disease is rampant in tropical and subtropical regions of South America, Africa, and also Asia. From the World Malaria Report in 2023, there were a total of 249 million cases that were reported of malaria, and out of the 249 million cases, there were 608,000 deaths. Africa remains to be the continent that carries the biggest burden of malaria, with 94% of the malaria cases reported happening in Africa, and over half of the deaths happening also in Africa, with 4 countries being particularly affected, which is Nigeria, Democratic Republic of Congo, Tanzania, and Niger. Currently, we have 3 measures that we use to control and prevent malaria. The first one is through the use of insecticide-treated nets. The second is through vaccines, and the third is through drugs. Each comes with its own challenges. For insecticide-treated nets, we have a challenge of mosquito vectors that have become resistant to insecticide. Of the 88 malaria endemic countries, 78 countries have already reported mosquito resistance to at least one insecticide. With the use of vaccines, we have the RTSS vaccine, which was approved by the World Health Organization in the year 2021, which has been really helpful. So far, they have reported a 13% decrease in early childhood deaths. Nevertheless, the vaccine has also reported adverse effects on the people that it's administered to. For example, for children, there have been reports of them getting cerebral malaria, malaria in the brain, and also there have been increased mortality of pregnant women who have received the vaccine. So the current mainstay of method of treating and preventing malaria remains to be drugs, and artemisinin-based combination therapies are the current drugs that are widely used in Africa, in South America, and also in Asia. Artemisinin-based therapies combine two types of drugs, that is artemisinin and their derivatives, which have a short half-life, and long half-life partner drugs, for example, amodiaquine, piperaquine, and lumefantrine. Artemisinin, artemisinin and their derivatives have a short life because they're able to clear the parasite very quickly, but they're dispensed out of the body just as fast, while long half-life partner drugs clear the parasite slowly and gradually, but stay within the bloodstream for 16 days or more, depending on the type of drug. So we chose to look at transporters because they play a vital role in the parasite's functions. If, for example, they transport nutrients, metabolites, waste, and even drugs. And because of this function, they are perfect mediators for drug resistance. So they are able to mediate drug resistance through, if they are a target for the drug, they can get mutations in order to evade the drug. They are also able to reduce drug levels if they are transporting the drugs themselves. And additionally, they can create pathways to efflux drug out of their target site so that they do not reach a concentration where they can work. An example of a transporter that has been implicated in drug efflux is the Plasmodium falciparum chloroquine resistance transporter. So the hypothesis of the study was that the selected transporters do not possess non-synonymous mutation that associate amodiaquine, lumefantrine, or piperaquine resistance in Plasmodium vagae. And our specific objective was to assess the stability of amodiaquine, lumefantrine, and piperaquine resistant Plasmodium vagae parasite using a standard 4-day test. And the second objective was to identify non-synonymous mutation in selected transporters that are associated with amodiaquine, lumefantrine, or piperaquine resistance in Plasmodium vagae anchor parasites. So these were the experimental approaches. The first thing we did was to reduce the number of transporter genes that we work with to a plausible number. So we did a blast analysis on PlasmoDB, and we used certain criteria to narrow down to a workable number. So there are a total of 145 genes that code for transporter proteins, but we were interested in those that were associated with membrane transport, which narrowed down to 96. We are also interested in genes that we could be able to knock out or overexpress, which would lead to dispensable or slow growth phenotypes, which led to 44 genes. And then we were interested in genes that were expressed in the asexual blood stages, which is the most active, metabolically active stage of the parasite, which narrowed down to 29. And then from the 29 genes, we did a multiple sequence alignment with Plasmodium vagae orthologs, 5 to be specific, to look for genes that were well conserved across the different species, which led to 9. And from the 9 genes, we also looked at genes that were associated with gene families that had already been implicated in anti-malarial drug resistance. So we started with 4 genes in our work, and these genes were core A, like magnesium ion transporter, which is MIT2. We also had ABC transporter G, family member 2, which is ABCG2, nucleotide transporter NT2, and novel putative transporter 1 and PT1. So the first preliminary test was to assess the drug sensitivity profiles using a 4-day suppressive test. And this test was to just confirm that our parasite had remained stable because they had already been stored at -80. So we infected mice with blood, red blood cells that had the parasite, and then allocated them into random groups. We had infected them with amodiaquine-resistant parasite, piperaquine-resistant parasite, and lumefantrine-resistant parasites. And then we had control groups that received no treatment, and then we had another control group which only had the wild-type parasites and not the resistant one. We treated them from day 0 all the way to day 3, and on specifically day 3, we took slides to look at the parasitemia and how the parasite was growing. And then on day 4, day 7, and day 9, we also took slides, but notably on these days, we did not treat the mice with their respective drugs. So we assessed the drug profiles by counting the parasitemia, and then we created these profiles. So the profiles generally reveal stable amodiaquine, lumefantrine, piperacin resistant Plasmodium buggy parasites. So from the line graphs, the green lines in each of the lines represent the wild-type parasite, which grew as expected, and the gray lines in each of the graphs show the resistant parasite to each respective drug, which also grew as anticipated. So in the first graph, graph A, we had parasites that were treated with amodiaquine, which are the amodiaquine resistant parasite. And the wild-type parasite was treated with a low dosage of 5 milligrams per kg, and the growth was suppressed, which was anticipated, while the resistant parasite, which was treated with 4 times higher of the drug, which was 20 milligrams, continued to grow, which shows that it retained its resistance even under storage. This was the same case with the lumefantrine resistant parasite, and the same case with the piperacin resistant parasite, where we can see that the wild-type parasite, the growth was suppressed, while the resistant parasite, which was treated with the highest dosage, continued to grow, which is the blue line. After ascertaining that our resistant lines were stable, we did a DNA extraction, a PCR amplification, and then sequencing. And then we looked at the data to check whether there were any mutations that were found. This is a summary of the data that we found. In MIT2 gene, at the gene level, we found a deletion of the base 19 at position 1234. And this at the protein level resulted to a premature stop codon being introduced at position 433. We also found an non-synonymous mutation in NT2, which led to another non-synonymous mutation at the protein level, which was a change from aspartic acid to histidine at position 511. And at NPT1, we found in piperacin parasites, we found a 255 change, an adenine change to a cysteine change at position 255. But the resultant amino acid code did not change, so there was no change in the protein. These mutations in MIT2 was specifically in amodiaquine resistant parasite, and in NT2 and NPT1 was specifically in piperacin resistant parasite. Lumefantrine resistant parasite contained no mutation in our genes of interest. So we wanted to assess the impact of the mutation in the MIT2 protein, the deletion mutation in MIT2 protein in amodiaquine resistant parasite. So the mutation resulted in the introduction of a premature codon at position 433, and this led to a truncation of the protein. A sizable part of the protein was cut off, and what we noticed was that some of the ligand binding residues that were in the wild-type parasite were lost in the resistant parasite. To check whether the protein structures in the wild-type and the amodiaquine resistant parasite were different, we did a superimposition of both structures to look for the root mean square deviation, and the score was at 2.734 angstrom and a Q-score of 0.002. So context, the threshold to check whether structures are similar is that they should have an RMSD score of less than 2 and a Q-score of more than 0.4. So from our analysis, we could see that the structures were very dissimilar, and we hypothesized that there could be a disruption in magnesium transport because of the loss of the ligand binding site, which would disrupt the protein's ability to attach to its ligand or the molecules that it was interacting with before. Okay. So the non-synonymous mutation in NT2 protein in piperacin-resistant parasites had no effect in the resultant structure. So the aspartic acid at position 511, we could see in the piperacin-resistant change to a histidine, and we superimposed also the two structures in wild-type parasite and piperacin parasite, and we got an RMSD value of 1.164 angstroms and a Q-score of 0.745, which ascertained that the structures were similar. We also looked at the hydrogen bonding between the amino acids that interact with the aspartic acid at position 511 and the amino acids that would interact with the histidine at position 511, and it was ascertained that the hydrogen bonds were not disrupted and that the amino acid in each of the parasite interacted with the same amino acids. So there was no disruption at all. So a summary of the study is that amodiaquine, piperacin, and lumefantrine selection pressure on Plasmodium vagae in vivo induces varied mutations, specifically in MIT2 and NT2. And these are non-traditional transporters which have not been studied before. And we show that the mutations in MIT2 led to significant structural changes. The deletion of MIT2 carries significant structural and functional implications for drug-resistant Plasmodium vagae parasite under amodiaquine pressure. And the aspartic acid 511 histidine mutation in NT2 did not carry significant structural and functional implications for drug-resistant Plasmodium vagae parasites under piperacin pressure. So from our table analysis, we saw that the ABCG2 and NPT1 proteins remained unchanged despite the selection pressure from amodiaquine, piperacin, and lumefantrine. So whether these mutations are linked with mediating drug resistance or mediating drug susceptibility would require experimental validation. Thank you. I would like to acknowledge Kemri. This is where I did my work. The people from the Center of Biotechnology Research and Development, also the people from Center of Traditional Medicine and Drug Research. I would also like to acknowledge my co-authors from Pan-African University, Dr. Beatrice Ashie and Ms. Jean Chetnitich, and my supervisor from JPAT, Dr. Daniel Kiboi. Thank you.
Thank you so much, Dr. Brenda, for that wonderful presentation, giving us insight into the stability of amodiaquine, lumefantrine, and piperacin in
Probably there was some internet disruption. Let us wait 1-2 minutes. Maybe so we don't lose the time, I would like to ask the next speaker, Dr. Puya Bartvai, maybe to introduce yourself and start your presentation. Thank you.
Thank you, Tanya.
Thank you.
Actually, I am not able to share my screen. I was saying that the other participant is sharing.
Yes.
Sorry for the network interruption. I would like to introduce the next speaker, Dr. Puja. Dr. Puja.
Yes.
Dr. Puja is a scientist at the ICMR Regional Medical Research Center. And can everybody hear me, please? Can you hear me, please?
Yes, yes.
Yes, yes. Now
Yes. Dr. Puja works as a DHR young scientist at ICMR Regional Medical Research Center in Gorakhpur. She completed a doctorate in biotechnology from RTMNU in Nagpur in 2020. However, during her PhD, she worked as a CSIR NERI in Nagpur. She worked on bioremediation of atrazine-contaminated soil using a combined approach of conventional microbiological tools and molecular tools with bioinformatics. She used genomics and metagenomics approaches to study the functional pathways and microbial community in pesticide-contaminated soil. Our previous research interest was on bioremediation through multidisciplinary approaches, whereas our current research area include the field of translation research with the main focus on diagnosis and post development. Our research interest employs advanced molecular tools such as RPA, LAMP, and CRISPR for the development of POCT to facilitate diagnostic in remote areas. She uses molecular techniques and bioinformatics to trace the evolutionary pattern and phylogenetic relationship of pathogens such as the dengue virus and orangia virus. She has been a member of the Organization for Women in Science for the Developing World, OWSD, since December 2019. And the floor is yours, Dr. Poja, and you have just 12 minutes for your presentation. Thank you.
Thank you, Dr. Olenke. So, greetings from India. First, I would like to congratulate the organizers for organizing such an applaudable conferences. Second, thank you for giving me this opportunity and a platform to be a part of this WFWS 2024. So, I believe we are talking about the health, and I believe this Sustainable Development Goal 3 depends on having a diagnosis and cure of all the diseases available. So before going forward, I, Dr. Puja Bhardwaj, would like to introduce to the disease scrub typhus. What is scrub typhus, which is also known as bush typhus? Why? Because this disease is contracted to the people who were living in close proximity or exposed to the scrubby or bushy vegetation. Scrub typhus is an important tropically neglected re-emerging vector-borne disease. Why I have said re-emerging? Because this disease has been historically reported or described during 3rd century from China, followed by 18th century in Japan, and during earlier 19th century, it has been reported from different parts of the countries, including India and Indonesia. Scrub typhus is an acute febrile illness which is transmitted by the bite of a Leptometridium mice at adult stage, specifically tigers, which is infected with Orantia shishugomati. And Orantia shishugomati is an obligate intracellular parasite. Why this name has been given to this pathogen? Because in Japan, Shushuga means insect and Mushi means illness. So the illness which is caused by insect is Shushuga Mushi. Also, this disease has been confined to area which is known as a physiognomatic triangle. Hence, the disease, the pathogen is named as oriented physiognomatic. For this disease, primary host is rodent, and mice is both a host and a vector, whereas humans are the accidental host. So, as I have said, this has been earlier confined to its region, which is known as physiognomatic triangle, which is Pakistan to the northwest, Japan to the northeast, and northern Australia to south has now been extended from its endemic region. The good thing about the disease is antibiotics are available and are highly treatable during early stage. But if diagnosis happens at the right time, but if this didn't happen, the delayed diagnosis can lead to acute encephalitis syndrome or multiple organ dysfunctions or case fatalities or severe mortality with a rate as high as 30 to 70%. This is the global data that it can cause mortality up to 30 to 70%. So this is the map you can see. This is the physiognomatic triangle, and the disease has now been globally reported from this different part of the country. Talking about the India, leaving 4 to 5 states, scrub typhus has been reported from almost every state of the country. So what is the cause of concern if treatment is available? As I have said, this is a disease of area with scrubby vegetation, means rural area or semi-urban area where diagnostic facilities are not there, or if available, infrastructure is not to the mark where diagnosis can happen at a time. So in India also, scrub typhus resurgence has been reported from different geographical regions, as I have already showed you in the map. As I have already said, it's a common cause of AFI and important cause of AS. Why important? Because from different states of India, like from Assam, it has been reported that 20% of the AS cases were due to scrub typhus only. And from the eastern Uttar Pradesh, 60%, more than 60% of the AS cases are contributed because of the scrub typhus only. So, without the appropriate treatment, I have said that treatments are available, but without that, case fatalities can reach to 50% or higher. And why? Because of the underreporting of this disease. This disease has been long a tropically neglected disease, and there are no rapid diagnostic tests available in the country to facilitate early detection of this pathogen. So what is the status of scrub typhus? There are the 4 ways by which scrub typhus can be diagnosed: direct microscopy, culturing, and both of these techniques require BSL-3 lab facility, which cannot be possible in the rural setting. Third is the serological method, which includes field select, immunofluorescence assay, or ELISA assay. These tests can provide a good diagnosis, but at a later stage, or you can say when a person gets infected with the disease, and 7 days, more than 7 days, it takes antibody titer to level to rise. So it takes a long time for getting diagnosis. And suppose if a patient reaches to the hospital during early course of infection, the patient may get misdiagnosed. And fourth is the molecular. For molecular, we are having PCR, conventional PCR, nested PCR, real-time PCR. These are the gold standard techniques, but again requires a sophisticated instrument and a trained personnel, which was not feasible in the rural or remote setting. The last option left is the rapid diagnostic kits based on the molecular, which are not available in India. So we aim to develop such nucleic acid-based RDT to promote early detection of orange hair physiognomatic. Now, why we have taken nucleic acid? As I have said, for the serological method, there are number of RDTs were available. But again, the rise of antibody titer takes more than 7 days to get to the detectable level. Again, this serological test also deals with the low sensitivity and cross reactivity. And as I have already said, this disease, once get delayed, can lead the patient to enter into the phase of severity. Whereas nucleic acid can be detected right from the day of onset of fever. These are more sensitive and specific over to serological methods can enable early detection of Orange schistosomiasis, but the bad thing is that such kits are not available as a POC. So the question comes in mind is how the nucleic acid amplification is possible to carry out at a room temperature or an ambient temperature or a single temperature. As in a thermal cycler, we need a different temperature to amplify the DNA or RNA or in a common genome. So in recent past decades, a lot of isothermal techniques have come into work, such as LAMP, RPA, CSR, among which recombinase polymerase amplification, that is the RPA, has showed its superiority over the other in its simplicity of primer designing and of course elimination of the sophisticated instruments, as it can be performed at a slightly higher ambient temperature, that is 39 to 42 degrees Celsius, and within a 10 to 20 minutes you could have a millions of copies generated of the target gene. So before the development of the assay, the first question that comes for the molecular detection is what would be the target gene, as the success of the molecular test or molecular kits depends on the successful selection of the target gene. So we have selected a range of genes: 47 kDa, OEL, 16 RNA, 56 kDa, and 27 kDa, which has been reported for the diagnosis of Orange schistosomiasis globally. And what we have found in our result that 56 kDa has demonstrated its outstanding performance in molecular detection, as we have taken a total number of 100 samples, among which 59% were IgM ELISA negative for scrub typhus and 41% were IgM ELISA positive. And among that IgM ELISA negative, this gene has detected the genome of scrub type or Orange schistosomiasis in 64% of the samples. So you can have an idea from this that what is the sensitivity of ELISA or serology test, that we are getting the 64% positive in IgM ELISA negative result. And also we have confirmed this pathogen by sequencing, and we have found that William and Carp are the predominant genotype of Orange schistosomiasis. And from this part of study, we have came out with these two publications. So now we came to the main part, that is the development of the assay for scrub typhus detection. So this is the strategy that we have adopted, which is divided into two parts. First is preparation of the positive control. Second is development of isothermal LFA is lateral flow-based detection, as it is the simplest way that can facilitate endpoint detection and can be read out by a normal common person or a technically unskilled person. This endpoint test can be read out. This is the complete principle on which this assay works. So here we have isolated the DNA from the pathogen, then we perform RPA. So this RPA recombinase polymerase amplification by using enzymes recombinase and polymerase, and by using some single-stranded binding proteins can facilitate amplification of the genome at a isothermal temperature. After getting the target genome, this genome has been detected by a CRISPR-Cas12a. You might have heard about the CRISPR-Cas9, which has been widely used for the genome editing. And these are the parallels of the CRISPR family only, Cas12a. And its activity of cis cleavage and trans cleavage that I can explain. Cis cleavage is upon a target recognition, means with the help of the CRISPR RNA or guide RNA, this CRISPR-Cas endonucleases recognize its target, cleaves it, and this is termed as cis cleavage. And upon target recognition, what it does, any single-stranded DNA present in the surroundings, it also started cleaving them. And this activity of this is termed as transcleavage. So with the discovery of the cis and transcleavage activity of this CRISPR-Cas12a and the nucleus, this has been employed for the detection of targets. And this reporter can facilitate the endpoint detection to amplify the signal. So this is the way by which we interpret the result. If the target is present in the sample, target gets cleaved, the reporter gets cleaved, and we have in the lateral flow state biotin ligand. This biotin ligand gets captured at the test line, and if target is present, this gets cleaved, and we do not get any signal at the test line or a very faint signal at the test line in the positive result. But in case the target is not present, the reporters get captured at the T line, and we get a broad colored test line and a faint or a broad colored control line, as you can see here in the result. So this is the overall principle that works. So these are the results. We have successfully cloned the complete ORF of the gene 56ADA from the two prevalent strains that were circulating in India, Gilliam and Sharp. And on the basis of this, we have designed 3 primers for recombinant polymerase amplification. And among the 3, we have selected this pair of primer. And on the basis of the product that is amplified with this pair of primer, we have designed 2 guide RNA or cRNA which facilitate CRISPR-Cas detection. As the designing of the cRNA also depends on the presence of the PAM sequence, that is triple TG in case of CRISPR-Cas12a. And further, we have among these two, we have found that CRISPR RNA2 has given the good result. And we have also confirmed the cis and trans cleavage activity with the gel image, as you can see, in the presence of the Cas12a and cRNA2, this the target DNA gets debated. Further, we have also optimized the concentration of this enzyme, cRNA, and reporter to get the optimal result in the minimum concentration of this treating. So what we have got the concentration of 30 nanomole for Cas12a endonuclease and cRNA, and 50 nanomole of single-stranded DNA reporters can provide the optimal results. And we have prepared a panel of 81 samples, among which 43 were positive by PCR for Orangeria schistosomasi, 38 were negative for Orangeria schistosomasi, and Rickettsia, which is a closely related pathogen to Orangeria schistosomasi, we have taken. And what we have found is, among this 43 positive sample, only 1 sample was falsely negative detected by this developed assay. So this platform has, these are the key features of the platform, that it works on the isothermal amplification, and it provides the endpoint detection with ease of interpretation by district. And by the panel with the clinical validation, we have seen that at laboratory, this has demonstrated 97.6% sensitivity and 100% specificity. This can be readily deployed at low resource settings or remote areas, and the limit of detection that we have found for this developed assay is 1 gene copy of the target. And by comparing with the other reported literatures, what we have found, this is the first study that has been harnessed the CRISPR-Cas12a enzyme for the scrub-typus detection. And of course, the incorporation of this enzyme for detection person has led to the sensitivity, superior sensitivity, I have to say, that 97.6 and 100% specificity, and limit of detection to 1 gene copy, which is higher than the other previously reported literature. So what, once this assay can be assay deployed at the field, what we expect from this assay is that it provides prompt treatment to the disease, as it's a positive agent of acute febrile illness. So it puts clinicians in dilemma that which treatment has to be started when patient arrives to them. as they don't have any peculiar symptoms to the disease. So it will help clinicians. It will further help in the reduction of case fatality and morbidity, as the disease, if get early detected, treatments are available. It will help in the upliftment of the health status of the population, and it will help in strengthening of public healthcare settings, research labs, surveillance studies, and it will also enable national and global public health security and mobile diagnostic units. It can be deployed. So the main limitation, which comes while the deployment of this AC to the field, is the instrument-free nucleic acid expression step, as this step requires a centrifuge-like instrument. So this is the limitation of this, and we are working on it. And maybe this year or the next year, we will be having that method in our hand to have an instrument-free nucleic acid extraction. And from this study, we have published this.
Dr. Fogia, your time is up. Thank you so much for that beautiful presentation.
And I would like to acknowledge all the persons
involved. Dr. Fogia, thank you. Your time is up. Let's give time.
The last slide only. Thank you.
Okay, quick.
Thank you.
All right, thank you. Thank you so much for that presentation. We are still awaiting questions and answers from the audience. Please, if you have any question, please drop it in the Q&A session, which will be tackled immediately after the whole session. Now, I would like to go to the next presenter, with Dr. Hayat Sama Mohammed. Dr. Hayat Sama Mohammed is from Giza in Egypt, and she graduated from the Faculty of Science in Hassan Sharma University at year 2017. She has a master's degree in bioinformatics at the Faculty of Science, L1 University, and she has published in peer-reviewed journal. So Dr. Aya will be presenting on bioinformatics approach for the prediction of analysis of non-structural protein 4B of Zika virus. Now the floor is yours, Dr. Hayat Sama Mohammed.
Hello, pleased to meet you in this greatest conference, World Forum Women in Science, online conference, Envisioning Tomorrow Science for the SDGs and the new partnerships for sustainable future. I'd share screen. Presentation on the title Bioinformatics Approach for Prediction and Analysis of Non-Structural Protein in SB4B of the Zika virus. Zika virus moved outside Africa, first spread to Uganda in 1947. Secondly, in 1960 to Nigeria. Third, in 1970 in Malaysia and Indonesia. In 2007, spread in island of Yap, Micronesia. Then in 2013, epidemic on French Polynesia. Zika virus infection, it transmitted via mosquitoes into humans. Pregnant women transmit the virus to fetus, causes microcephaly. Zika virus organization, genome structure, it consists of 3 structural proteins which form virus particles and 7 non-structural proteins. The target protein is in S4B. In S4B induces the formation of endoplasmic reticulum-derived membrane vesicles where viral replication takes place. NAC4B protein of Zika virus of 251 amino acids was retrieved from UniProt KB, which is the aim in this study. Protein structure prediction, it is a proteomic tool for understanding phenomena in modern molecular and cell biology. Has an important application in biotechnology and medicine. One of these applications is the prediction of three-dimensional structures of virus proteins, hence elucidating their function. This will aid in drug design and integrative understanding of viral processes. Classes of the protein structure. First, primary structure consists of amino acid sequences connected together by polypeptide chains. Secondary structures consists of alpha helix and beta sheets connected together by loops. Tertiary structures folds and bags upon itself. Assembly and folds upon itself. Quaternary structure interaction of two tertiary structures. Three approaches of protein structure prediction. First method is the template-based modeling, homology modeling. It is called comparative modeling. It is to compare similar templates on the database with the target sequence. Second method is fold recognition, is to compare similarity between structures, ignore similarity between sequence, which need to make secondary structure prediction. Template-free modeling, is post ab initio modeling, which is to compare the coordinates between residues. Then collecting all residues, all similar residues, ignoring dissimilar and repeat the same method. Objective: 3D structure prediction of NSB4B by using servers and software according to case. Reduce level structure assembly and refinement simulation, model evaluation and selection, prediction and analysis of motifs and post-translation modification sites of NSB4B, functional analysis of NSB4B, structural classification, correlation of structural and functional prediction, material and method. Schematic view for protein structure prediction. Firstly, we have the protein sequence, make searching on the database, then multiple sequence alignment is made. Search homolog protein, which is called template. If similarity became greater than 30%, we make homology modeling to get 3D protein model. If identity less than 30%, we make secondary structure prediction, then fold recognition. Then if fold predicted, we make multiple sequence alignment, then homology to reach 3D model. If no, we make ab initio searching about the similarity of coordinates to reach 3D model. In our study, we made multiple sequence alignment and conserved regions using cluster omega servers and BioEdit 7.2 software. Phylogenetic analysis using cluster omega servers, Mega 11 software. Domain separation using 3DOM, ProDOM servers, and NCBI conserved domain search. Secondary structure prediction using Proteus 2 server, NBS at Sigma, Raptor X, and Predict Protein server. 3D structure prediction using iTasser server, SwissProt, Galaxy Web, Lumens 3, AlphaFold, Colab. Model refinement, 3D refine server, Mod Refiner, Galaxy Refine servers. Model evaluation using save server, rocheck, watcheck, TM score, TM align, Q-mean, superpose servers. Functional motive prediction using Brozet, Scan Brozet, Smart, Scan Site, BFAM, Motive Finder, and Motive Scan. Structural classification using Interbro, Scope and Case servers. Results and discussion. First, multiple sequence alignment. Multiple sequence alignment of NSB4B with most homolog protein using bioedit 7.2. It is the target protein, non-structural protein NS4B Zika virus, which it is made alignment with all similar proteins, all with the polyprotein Zika virus, similar regions. Similar regions from 2270 to 2520 conserved regions. Conserved regions, 4 conserved regions were predicted segment length 22, 41, 81, and 57 with average entropy approximately 0. Maximum likelihood estimate of substitution matrix. It is a table of substitution matrix. Column and row represent 20 standard amino acids. Each one changes together at frequency of time. Each change represents a matrix. Then full profile or Markov model is made, which is the algorithm to make phylogeny. Two methods of phylogeny use the maximum likelihood method, which made this figure as non-structural protein similar to all ancestors, all of which polyprotein Zika virus. And it is the second method, neighbor joining method, which made the same result of maximum likelihood non-structural protein similar to polyprotein Zika virus. Domain separation. One domain from position 4 to 190 with cutoff value 0.66. Secondary structure and solvent accessibility prediction. Secondary structure prediction using Sigma server alpha helix with percentage 54.58. Extended strand 12.35, beta tail 8.37, random coil 24.70. Solvent accessibility prediction using predict protein server. Exposed regions 35.46, intermediate 2.79, buried regions 64.54. 3D structure prediction. The top 5 models were predicted by ITASR using the confidence score of each model, which is typically in the range of -5 to 2 on the C-score and the protein length. TM score was calculated to be 0.34 plus or negative 0.11, and RMSD to be 14.1 plus or negative 5.8 angstrom. Based on a confidence score, AlphaFold built 5 models according to predicted local distance difference test. It shows a very high confidence score with BLDDT greater than 90, confidence greater than confidence score between 70 and 90, low confidence between 50 and 70, and very low confidence BLDDT less than 50. The best one was with BLDDT score 83.8 and BTM score 0.74. Reduced level. The original model produced by ITOSR servers, reduction of RMSD from 14.1 to 2.7 and increase of the TM score from 0.34 to 0.77. Also, in refinement, AlphaFold structure increased the TM score from 0.74 to 0.96, which led to a notable improvement in the physical quality of both global and local structures. Evaluation of predicted 3D structures according to CAD. The best results were from ITASR and AlphaFold collab. The best results in this research were ITASR, but AlphaFold collab was predicted as the best model in CAD 15 according to paper Jumper et al. 2021. Ramachandran plot. We have 70 core regions, 77.2%, allowed regions 16.4%, generously 3.2%, and disallowed 3.2%. Residues in most favored red regions B, A, and L are 169 with 77.2% residues in additional allowed regions. Yellow regions A, B, L, B are 36 with 16.4%.
So, Dr. Aya, please, can you please round up because of our time? We still have two presenters, and so
okay, I will
time for questioning and answering.
This template was from ITASR for CE 7A is the accession. RMSD 3.36, 3.36, TM score 0.77, aligned length 241. Signatures and motif analysis. Two motifs. One first is the flavivirus non-structural. Is 66 to 82. Post-translation modification sites. 4 hits with 11 active sites, which is used to make docking for
your time is up, and we will let's take the next presenter because of our time. Thank you so much for your presentation and the knowledge you have given unto us in the prediction and analysis of non-structural proteins for Zika virus. Now, the next presenter is myself. I happen to be the moderator and also one of the presenters. My name is Olanike Christiana Akinduite. And Dr. Aya, please, can you just unshare your screen so that I can go ahead with mine?
Okay.
Okay. So thank you. My name is Dr. Olanike Christiana Akindite. I'm a computer scientist and lecturer at the Department of Computer Science at Tafe Babalola University, Ado-Ekiti in Nigeria. I have a co-researcher with me on this research, the research on sepsis, who is a computer scientist too, who is Dr. Bukola Badeji Ajisafe at the same department. Our research focus is in the area of machine learning, where we are leveraging on the effectiveness of machine learning in predicting some diseases in order to 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 improve patients' outcomes and to prevent the insurgence of the disease. And one of the diseases in our research is in sepsis. And that brings me to the topic of understanding sepsis, causes, symptoms, diagnosis, treatment, and prevention strategies. One of the 17 Sustainable Development Goals is good health and well-being. And all the 17 Sustainable Development Goals are integrated. That is, they recognize that action in one area will always affect the outcomes in others. So therefore, no matter which one of the 17 goals that we are aiming to achieve, is it
طب
to
الكور CE سين A was accession number protein
padding with. So we realize that good health and well-being is central to all to all the 17 goals. So realizing SDG 3, which falls under health, determines the progress in other SDG according to WHO. And the word under discussion is sepsis. If we look at the cloud of words surrounding sepsis, all the words surrounding sepsis, they are not friendly. We have death, we have infection, virus, bacteria, organ failure, shocks, severe time sensitivity. This brings us to the awareness that this disease called sepsis is life-threatening, is dangerous, is severe, and there is a need for public awareness to bring to the knowledge of the populace that the sepsis can result into death if not uh, uh, if proper knowledge is not given at the proper time. Now, what is sepsis? Sepsis has a lot of definition according to different authors, but one thing is central to all the definitions. That one thing is central is a serious condition of health, is life-threatening, is caused by infection, and is a medical emergency that is caused by a body's overwhelming reaction to infection. And most of the time, it can lead to damages in our tissue, it can lead to organ failure, it can lead to death. So all these definitions point us to the fact that sepsis is a dangerous disease and is not common among people have not come to the realization of the presence of sepsis and among humans. And now, what are the stages of sepsis? We have different stages of sepsis. We have ordinary sepsis, which is as a result of the systemic inflammatory response syndrome, which we call SARS, which ordinary happens to surface when there is high body temperature, high heart, when the heart rate is beating fast, when the blood pressure is low or is too high. Those are the normal signs and symptoms of SARS. But when any of these signs and symptoms is present with an infection, then there is occurrence of sepsis. So the first stage of sepsis has to do with an infection on the normal signs of systemic, the systemic inflammatory response and syndrome. And the second stage of sepsis, we call it severe sepsis. Severe sepsis is when the infection and the inflammation becomes acute and begin to gradually affect some organs like the liver, the kidneys, the heart. And when this begins to happen, there's possibility of organ failure. And the most serious stage now is the third stage, which has to do with septic shock. This stage refers to a serious complication of sepsis, resulting in a significant drop in blood pressure. At this time, of course, there is permanent, there is total organ failure, and the respiratory or heart failure has occurred, and there is possibility of stroke, and in some cases, most of the times it results into death. And now, what is the importance of understanding the word sepsis? Globally, there are report cases, according to WHO, that the estimate for 2017 shows that sepsis affects up to 50 million people, and it causes 11 million deaths yearly. So it has been reported that is one of the leading causes of maternal that affect the women, mostly during pregnancies, and neonatal and child mortality rate, which has contributed to over 40% mortality rate for patients, most especially in the critical care unit. And according to the Centers for Disease Control and Prevention, it has been reported that this dangerous disease called sepsis is a major reason people who are discharged from the hospital are readmitted back. In fact, there is a report that most of the people that are discharged has the high risk of returning back to the hospital within 30 days of their discharge, and they are readmitted back to the hospital. So anyone can get an infection like COVID-19, but when an infection is present, it leads to sepsis. So in a typical year, we have an average or at least 1.7 million adults in America that develop sepsis. And out of this 1.7 million, like 350,000 adults who develop sepsis died during hospitalization. And 1 in 3 people who dies in the hospital has sepsis during that hospitalization. So combating sepsis will help us to contribute to the achievement of sustainable development goals on the quality of healthcare according to WHO in 2023. Now, what are the causes of sepsis? Nearly any infection can lead to sepsis. It can be bacterial, it can be viral, it can be parasitic, it can be fungi. But most of the times, the germ that is mostly responsible for causing sepsis is bacterial infection. And unfortunately, not all of these pathogens, not all of these germs can be cultured. So there is this calls for a need for continued awareness of the presence of sepsis, especially in the hospital. Now, what are the conditions that can cause a higher risk of causing sepsis? People that are affected with pneumonia, and people with abdominal infections, people with kidney infections, people that have blood poisoning already, or people that are treating open wound, they are at higher risk of contracting sepsis. And the risk factor that can lead to sepsis are those people that are aged, when people that are above like 65 years of age, or for people that has an increase in antibiotic resistance, or people with an increase in the number of people with illnesses that have a lot of diseases like diabetics, like improper breathing patterns, that weaken their immune systems. What are the symptoms of sepsis? Like I said earlier on, general symptoms like high body or temperature or low body temperature, high IR rate or high respiratory rate or high or low white blood cell count. They are normal signs and symptoms of general body illnesses. But there is the presence of systemic inflammatory response syndrome, called SARS, which shows signs, all these signs. But one thing is paramount, and that is when any of these signs, one of these signs is present, and the presence of an infection is confirmed, then there is uh, sepsis. So depending on the stages of infections, one may be diagnosed with sepsis if you have a probable or confirmed infections alongside all these signs that I've mentioned, and as well as any of the following signs like a high rate, high heart rate, high temperature, high respiratory rate, confusion and disorientation, which has to do with mental instability, slow speech, and the likes. And now, the symptoms of sepsis has to do with stages. For stage one, at least one or even more of those signs has to be present. And for for people living with sepsis, for stage two, it would have progressed to difficulty in breathing. It would have progressed to bluish discoloration of skin that when pressed on the, when that signs on the skin is pressed with finger or with anything, it doesn't disappear easily. There is presence of dizziness or reduced urination, extreme changes in mental ability, extreme fatigue and unconsciousness and abnormal heart function. And in stage three, when a sepsis enters stage three, it can quickly get worse and lead to what we call septic shock, which is a critical medical emergency and is described as life-threatening. Some severe sepsis and septic shock and symptoms share the commonalities with all the symptoms we have talked about, but one, there is a very low blood pressure. That means the septic shock signs as materialized. And I have on the screen all the signs and symptoms according to the stages of the sepsis. And now, who are the risk factors of people that can fall into this life-threatening disease? Sepsis can affect anyone, but there are some people that have a higher rate of contracting these infections. Individuals who are particularly vulnerable to this are people that are aged over 65 or children that are less than 1 year old, people that are suffering with chronic illnesses, people that has a recent surgery and invasive procedure just because they have been exposed to all these invasive devices, which may include breathing tubes and intravenous catheters, and people that are that works in hospitalization or in nursing home, and people that has experienced one trauma or injury. And how can we diagnose sepsis? Depending on the signs and the symptoms of sepsis, a patient, it passes through three stages: the clinical assessment, the laboratory test, and the imaging studies. The clinical assessment has to do with taking the medical histories and of infections on the patient and carry out a physical examination of some vital signs. And after this is done, the next thing an healthcare provider does is to ask for the patients to go for a blood test. A blood test is checked for complications like infection, like abnormal liver or kidney functions, or decreased amount of oxygen, and the likes. And depending on the clinical assessment, the result of the patient blood test, the healthcare provider may order such tests as urine test to check for bacteria in the urine, a wound secretion test to check for an open one, or for an infection, a mucus secretion test, a blood culture, a coagulation studies, renal and liver function test, and arterial blood gas analysis. And imaging studies is carried out in a situation where the healthcare provider will not determine the source of the patient infection using the clinical assessment and laboratory test. So in this case, an internal view of the patient body may be required, which may have to do with conducting the chest X-ray to view the lungs, or the CT scans to view the possible infection in the appendix, in the pancreas or the bowel areas, ultrasound to view infections in the gallbladder or ovaries, or MR scans which can identify soft tissue infection. And how do we prevent sepsis? Of course, a strict and accurate information for effective intervention is required, like I said earlier, to save life and to reduce healthcare costs and prevent long-term complication, and of course to prevent death. And some of the preventive measures are hand hygiene, like vaccinations, infection control measures, early detection and treatment, which has to do with our core area of research, where we are using machine learning and ensemble learning algorithms to build models that can help in the early prediction of sepsis. And now, how do we treat sepsis? Sepsis is a medical emergency, and when it comes to sepsis, time is of essence. So anyone that has the signs that we talked about has to seek for medical immediate medical attention, has to go for medical checkups and diagnosis, for appropriate antibiotics and antifungi, has to seek supportive care in the ICU. And until a cure for sepsis is found, early detection and treatment is essential for survival and limiting disability for survival, which are our core area of research as a computer scientist. Thank you. Now, before our time is up, we have the last presenter, who is Dr. Karuna. Dr. Karuna is the chairperson at John's Medical College in Bangalore, and she's a pathologist at Rainbow Children Hospital from 2019 to date. She possesses MBBS, DCP, MD, and PhD in medical law and ethics, and she's a fellow of FICP, and our area of our field of specialization is hematology, which has to do with anemia and the likes, and also in quality control, tropical diseases and infectious diseases like malaria and HIV/AIDS, tuberculosis, and the likes. So the floor is yours, Dr. Karuna. Please, you can share your screen.
Good afternoon. Are you all able to see the screen?
Yes, yes, yes. You can see the screen.
Okay. Shall I go ahead?
Yes, go ahead, please. Our time is almost up.
Yes. Okay. Good afternoon to all of you and the chair and my co-speakers. Because I'm the last speaker of the session, thank you for your patience. And I thank the organizers for giving me an opportunity to present in this forum. We have had genetics, we have had sepsis and all those things. Mine is a very simple thing. How to connect, collaborate and care for patients by using healthcare screening and education programs. This was done at Mitra Multispeciality Hospital, Bangalore, Karnataka, India. If we go to the next one, are you going to see the next one? Okay. You all know about the sustainable goals. I'm focusing on the goal number 3 and goal number 4. That is good health with education with partly number 17, where we have a partnership for the goal. So now looking at this one, in a regular hospital setting, combining health screen with education program is difficult and requires more manpower. Collaboration with similar organizations for healthcare education will help in scaling the program. We took only one disease, which is a non-communicable disease, diabetes mellitus. So the primary objective was to screen for type 2 diabetes to identify at-risk patients. The BP measurement was also done as part of checking the vitals and to educate the patients regarding the course of disease and the follow-up. Actually, the hospital started in the year December 2020. So the diabetes, which is the most common disease in the Bangalore, the non-communicable disease, we thought we'll screen for that one. So they thought from January 2021, we'll start the screening program. But by the time the pandemic hit, so what had happened was we divided into 2 phases from January 1, 2021 to June 30, 2022. The screening was done only at the hospital outpatient department. The period covered second and third wave COVID pandemic. Only who were willing to be screened and referred by the physician were included. The patients who were in the pre-diabetic range were counseled and followed up. By the end of June, we realized that the pandemic is stopped and we are going to be normal. So from July 1, 2022 to December 2023, the screening was extended by organizing camps at the rural centers and at the city centers. Help was taken from the educational organizations and Rotary Club to organize the camps. Health education was done through individual talks and information materials. As I told you, the diabetes is increasing at a global level, at a national level, and Bangalore is supposed to be the diabetes capital of Karnataka. And just to give a background of this hospital, it is in the midst of your IT sector like Infosys and Wipro, and you have the villages setting, Kyalasane and Babasandra villages, and Bomasandra industrial area. So you cannot say it is only rural, it's only urban, it's only industrial. It was the population was a mixed population. So it was a cross-sectional study, and only from the OPD, as I already told you, would be willing to be screened for diabetes, and the symptomatic patients referred by the physician. So the 3 tests we took was fasting, postprandial blood sugar levels, and hemoglobin A1C for all patients by standard method. The fasting glucose at 126 was taken at the cutoff for the diabetes, and A1C 6.4 was taken at the cutoff level. So during this period, though there was outpatient who was 8,253, the number of patients screened are only 682. That is, they're willing to get screened. That's only 8%. Among them, the female preponderance was slightly high. The breakup of the 682 were confirmed diabetic were 216, non-diabetic were 364, 102 were the pre-diabetic range. In the pre-diabetic range, we had 5.7 to 6.4 were the cutoff. So 102 patients were there. Below 40 was 38, 40 to 60 was 53, and above 60 was 11. So they were counseled for the diet, activity, and genetic predisposition. And they were asked to come up after 3 to 4 weeks follow-up. The mode of follow-up was mobile and WhatsApp message. At this point, I want to tell about an Asian Indian pre-diabetes study. Compared to other races, Indians have higher insulin resistance, poorer pancreatic function, and a greater risk of developing diabetes, highlighting the importance of early strategies for improving insulin sensitivity and improving pancreatic function in Indians to prevent diabetes and lower the risk of heart disease. The challenges in follow-up were the complaints was acceptance was better in above 60 years. They said, oh, I'm 60. Okay, I'll accept it. They were diabetic. The others, they were little unwilling. And also there was unwilling for lifestyle changes. By the time the pandemic set up, the work from home style has started. So they were unwilling to change their lifestyle. And in the rural background, the complaints food habit was also not so much. So that is why the difficulties were in follow-up of the patients. For the diabetic patients were now 216 in number. As per the requirement, treatment was given. The complications like chronic kidney disease, diabetic foot, cataract, they were all there and they were followed up there. During this period, we classified into 4 groups: pre-pandemic from January to March, April to June as pandemic, July to December as post-pandemic, and January to June as follow-up period. If you look at it now, from the pre to pandemic, post-pandemic and follow-up, slowly the number has increased because people realize that now what is the most common morbidity, which is diabetes, and the patients are willing to be screened because of the awareness of the comorbidities. So to summarize the phase 1 finding, 682 patients were screened out, which 102 patients were in the pre-diabetic range. There's a gap between knowledge of risk factors and the need for screening and referrals, irrespective of the population. Pandemic increased awareness of comorbidities, which resulted in increased number of patients for screening post-pandemic and follow period. And linking diabetes screening with referral to lifestyle program will help in awareness, we thought. So what has happened by the end of the May, we thought we can scale up the programs. So from July 1, 2022 to December 23, the screening was extended by organizing camp at the rural centers and at the city centers. Help was taken from educational organization and Rotary Club to organize the camps. Health education was done through individual talks and information material. During phase 2, the number of patients screened were 1,085. 88 patients were in the pre-diabetic range who required counseling for nutrition and lifestyle changes. These are the few photos from the camps. I will just skip this. They are all one at the rural center. One we connected to the Rotary, and these camps were held. And these were all educational programs, like how we treated about the diabetes, kidney disease, what happens in the kidney and all. So between comparing phase 1 and phase 2, January 21 to June 22 was the pandemic period, and July 22 to December 23 can consider as back to normal. Here 682 were screened, here 1085 were screened. Here we use fasting postprandial hemoglobin A1C, but here we use only fasting blood sugar. Hospital OPD in phase 2 camps at rural centers. Here only hospital staff. Here we used so many organizations came to help. Here counseling was done. Here we had education program, dissemination of education materials and all that. Then it was self-funding and supported by the organization. That supported by the organization also limited to the funding. So the outcomes were the difference in findings highlight the advantages of collaboration to scale the screening and health education program, but also helped in deployment of calculation to understand the cost of screening and financing in the rural setup. The challenges, one is the test method, because there we use so many, but here we use fasting blood sugar. Follow becomes difficult. In a rural setup, follow becomes difficult. And the cost of the program goes up when you are setting up different centers, camps, and all those things. Because you have the support of the organization, we were able to do that. And my Mitra participated in the Unnat Bharat Abhiyan program, which is a government program, because realizing the knowledge base of the educational institutions. So what is the projection? The following are planned for the future. One is mapping of donors and cash flows for funding for screening, mapping of technical assistance, analysis of operating methods of free healthcare, like you have to align with government center, CSR funding initiative, advocating for the allocation of more resources for non-communicable disease, including diabetes. The main challenge will be to identify and clarify the long-term funding to screen and sustain the program and align with government initiative. I'd like to thank my team members, Dr. Bharat, Dr. Manoj, and Dr. Bipin, lab staff, field staff, and the partnership with Institute of Management, Rotary Connect, JNC Trust, and government setup. Because of this, we were able to screen diabetes. This is where the partnership helps in the healthcare program. Thank you very much. Ma'am, I just want to tell you, I had to condense because the time was a bit limited. Thank you very much anyway.
Thank you so much for your presentation and the limited time that is given to you now. We have 1 or 2 questions from the audience, though has been answered privately, but we may need to discuss it on the platform. This one goes to Dr. Poga. Thank you, Dr. Poga, for your outcome recommendation that typhoid is low socio-economic disease and is better to improve the population hygiene hygiene culture instead of going through the disease cycle without having the ability to diagnose and treat. So Dr. Poja, what do you have to say to this?
I think she already answered in the comment. And unfortunately, I have to say that our time is up.
Okay. Please read the answer in the comment.
Yes, let me read the answer.
Yeah, for general consumption. For general consumption.
Okay, thank you. Yes, that's part in governmental program already. In spite of these cases, were often diagnosed with CT. The problem in Eastern up India that it's the pediatric population that gets mostly affected, since it's an etiology of AFI cases are often goes undiagnosed and get diagnosed when it turns to AES.
Oh, okay. Thank you so much. And there is an observation from Dr. Dalla on the presentation on sepsis, that sepsis is not defined as a disease. It's a serious condition that has to do with infection with lower immune response. Yes, from my presentation, I do state that sepsis is not a disease. So it becomes an emergency, a medical emergency, when an infection is present in any of the signs of the normal signs and symptoms. So is human body extreme reaction or responses to infections? That is when sepsis occur. And our immune system usually works to fight any germs, be it bacteria, be it viruses, be it fungi or parasites, to prevent the presence of infection. And if there is an infection, our immune system will try to fight it. So although most of the time with the help of medication like antibiotics, like antivirals, like antifungals, like antiparasitic, parasitic, depending on which germs is infected, which germs cause the infection. But for reasons researchers don't understand often, that cannot be established now, sometimes our immune system stop fighting this thing called invaders, and they begin to turn, and our body begins to turn on itself. So this signifies the start of sepsis. So sepsis is not a disease on its own, but is our body's reaction to our body's over or extreme reaction to the response of infection, to the presence of infection. Now, I have a question for Dr. Brenda. Dr. Brenda, are you there for us?
Yes.
Does your study cater for how to measure how harmful scrub typhus is? How harmful does your study cater for how harmful scrub typhus is? And how long does it last if there is an infection resulting from scrub typhus?
Sorry, did you say typhoid?
Does your study cater for how harmful scrub typhus is? And how long does it last in human body?
Scrub? I'm sorry, I don't get that part. Scrub?
Do we have anyone that want to contribute down to help Dr. Brenda into the study on scrub typhus? How long does it last in human body, and how harmful is it?
My study was on mice. So the parasite I was using infects mice. We used Plasmodium bagi as a model for Plasmodium falciparum. because they have similar proteins. So the study that we did used Plasmodium vagae as a model. So the parasite in humans, Falciparum, sorry, the question is how long it lasts in the human body or the drug.
Okay, thank you so much, Dr. Brenda. Our time is fast spent, and another session is about to start. We want to appreciate everyone for your participation in this session on health, and I want to believe that we have all garnered some insight into some cautionary measures that we need to take, especially in all the diseases that has been discussed, like sepsis, like the Zika virus, like the typhus, and also for us to spread this to the public domain in order to reduce the insurgence of these diseases. Thank you so much. We are going to end this session now, and another session is going to start in earnest. Thank you, everyone, and we appreciate you for your participation.
Thank you for your important contributions, and we are wishing you a nice day.
Bye.
Bye-bye.
Bye-bye.